Did you know that temperature, humidity, and electrostatic charge can significantly affect inhaler test results? In this video, you'll discover why environmental control is essential for accurate dose delivery and dependable pharmaceutical quality.
When pharmaceutical companies develop orally inhaled products, measuring the exact dose delivered by the device is the only way to guarantee patient safety. If the quantity of medicine varies by even a few micrograms, the device could fail to treat the patient entirely. This line graph shows the aerodynamic particle size distribution, which tracks the physical size of the medicine particles. We expect a smooth, predictable curve. But in the lab, this data frequently fractures into erratic, unpredictable measurements. When tests return data this wild, investigators usually look for a human making a mechanical error or a faulty flow control valve on the testing machine. Yet, in many cases, the human operator is executing the test flawlessly, and the machinery is perfectly calibrated. The actual variables destroying the data are localized environmental forces: the temperature, the humidity, and the static electricity of the air inside the room. Failing to control these forces means even the most precise testing equipment will generate false failures, jeopardizing the approval of life-saving medical devices. To understand how the environment sabotages the data, we have to look past the laboratory bench, pushing inside the nozzle of the inhaler itself. Localized temperature fluctuations directly dictate how fast the liquid propellants turn to gas, while also changing the viscosity and surface tension of the drug. This is a cross-section of a cascade impactor. If the metal inside this apparatus gets slightly too warm from the room air, the incoming liquid drug droplets prematurely evaporate before they can reach the collection plates to be measured. Humidity plays an equally critical role. The relative humidity in the air dictates the physical formation and size of the liquid droplets the moment they are released. Dry conditions are particularly damaging for powder-based drugs, where a lack of moisture causes powder clusters to stick together, a failure in deagglomeration that prevents the medicine from breaking apart in the air. Unchecked fluctuations in heat and moisture physically alter the drug dose before it ever reaches the sensors, rendering the final data invalid. The split-second discharge of a medical inhaler into the laboratory testing apparatus creates a high-velocity, violent physical collision. As thousands of drug particles rub together and separate at high speeds, they generate an intense electrostatic charge. This is a phenomenon known as triboelectrification. This generated charge polarizes the particles. Positively charged drug droplets are violently drawn to the negatively charged metallic walls, sticking permanently. But raising the relative humidity, this blue vapor, neutralizes the charge, providing a path to ground so particles flow freely. To ensure the drug reaches the sensors instead of the walls, testing labs must maintain a strict threshold of 40 to 60. These chambers maintain extreme precision, holding temperatures within 2 degrees Celsius and relative humidity within a 5% margin. The third solution is active static mitigation. Technicians must physically ground both the equipment and themselves, which often requires wearing specialized anti-static shoes to prevent charge buildup while moving. To verify the environment is neutral, labs deploy ionization bars and use handheld static meters to confirm the air is ready. By shifting focus from the entire laboratory to the immediate microenvironment, scientists eliminate costly out-of-specification errors. This ensures the resulting data reflects the true performance of the inhaler, not the limitations of the room.